Good afternoon. Thanks for the invitation to present some of our data here for the Forge Analysis Super Bowl. It's a privilege. It's something I have the opportunity to see every year, but first time I had opportunity to present. So thank you very much, and I hope we can contribute with some useful information. So the objectives of our presentation today is to first introduce some indicators of corn silage nutritive value. And from there, we will try to highlight the use and application of some of those indicators and then move towards a more practical discussion of strategies that could enhance the quality of those indicators. The presentation will not be necessarily in this order, but we will try to follow all those objectives as we discuss first fiber and later starch digestibility of whole plant corn silage. So when we think about high producing dairy cows, a question that always comes to mind is what holds cows back? And if you think about high producing dairy cows, some of those cows, they are eating 55, maybe even 60 pounds of dry matter every day. But the issue associated with that is, even though this high consumption helps with very high energy intake, unfortunately, those diets are digested on average 62% of dry matter, which basically means if we consider a 55 pounds intake, only 34 pounds are being digested. Even though this is a very good number, the variation in dry matter digestibility is very high. It's plus minus 15 percentage units. So in reality, depending on cow diet, forage level, quality of the forage produce, and so on, this number may vary from 26 up to 42 pounds of digested material per day. And this is of particular importance because for every pound of TDN, or total digestible nutrients, that an animal can have, it's supposed to have enough energy to generate 3.5 pounds of milk. So our ultimate goal is to make sure we drastically improve TDN of the diet and consequently improve milk production. If we try to tie that back to the nutritive value of corn silage, these calculations are based on an article by John Gazer, and it's a similar calculation performed by other researchers in order to fully understand which fractions of corn silage play major roles on energy intake. And if we summarize all those numbers, and you will see the square or rectangle on the top right corner, basically those are the two most important variables, and they're exactly the same number that are presented into this graph. If we consider NDF digestibility, here highlighted as NDFD, 18% approximately of the total TDN of a whole plant corn silage sample is coming from NDF digestibility. For starch digestibility goes even higher, is approximately 55%. So when we combine that, we can say that 75% of the energy value of corn silage comes primarily from NDF digestibility and starch digestibility. So what can we do to improve those two fractions and from there improve milk production by high producing dairy cows? So first, we need to understand which metrics could we use to evaluate those two parameters, fiber and starch digestibility. So for the sake of simplicity for this presentation, we separated into fiber and starch. So we go through fiber first, and the remaining portion of the presentation will be on starch. So if we think about the most common quality metrics used for fiber digestibility in the United States, regardless if we are discussing corn silage, other forages, or the TMR, they are all highlighted in this particular slide. So if you think about that, first we have the parameter in the first column, followed by a second column with arrows that are indicating what should we do to improve the quality of the material. Then after that, we have the number of samples that were utilized to generate those numbers. And last, we have the normal range for all these different parameters. Okay, and by normal range, what I mean is from all the samples that we collected for this particular survey, and I'm saying we, but in reality it was Dr. Shaver, basically he used 2/3 of the samples to mimic one standard deviation plus and one standard deviation less. So basically this is what we expect truly for those different parameters. So first, if we go through them, we have NDF, which obviously we measure for many, many years. And we already know that slightly less NDF may lead to slightly better quality of silage because, as you saw in the previous slide, NDF generates less energy than starch, for example. And typically when we increase starch, we reduce NDF by dilution in whole plant corn silage. But obviously, we still want a reasonable amount of NDF because that's what provides rumen function and so on. After that, we have two indicators that heavily affect the potential digestibility of, in this particular case, corn silage, but in forages as a whole. First, lignin, which is basically the indigestible fraction of fiber. And after that, we have UNDF, that stands for undigested NDF. In this particular case, we are highlighting the UNDF 240, which is the UNDF after 240 hours of incubation, either in the rumen or in rumen fluid in the in vitro system. Regardless of if you use lignin or UNDF, the lower the number is, more digestible fiber becomes. So regardless of which indicator you're using, and more the dairy industry now is using UNDF just because it's easier to measure than lignin, and some of the formulation models are incorporating that, is becoming more and more real. Okay? And I believe that as we reduce those, it translates into the next two parameters, NDF-D30, which is the ruminal in vitro or in situ NDF digestibility at 30 hours, and the TT-NDF-D, which stands for total tract neutral detergent fiber digestibility. This is not total tract as in the dairy cow total tract. This is a prediction generated by 3 time points in the lab, 24, 30, and 48 hours, plus an indigestible measure such as the UNDF 240. So basically, less NDF, less lignin, and less UNDF, but greater NDF-D and greater TT-NDF-D is what we want to see. Just a last comment for this slide, you will see that the numbers for lignin and UNDF, they're very different. And this is very important because if we start using UNDF now, some people may be confused and go there and say, well, but I don't remember those numbers being so high before. But in reality, it's because the UNDF accounts not only for lignin, but also a couple other fractions of the NDF that are associated with lignin and not digested. But Luis, you just show all those different indicators, but you know, what do they really mean for a dairy cow? And if we put that into a dairy cow perspective, all the time you have more fiber, more lignin, more UNDF, or lower NDF-D and lower TT-NDF-D in your diet, you have a potential intake limitation because of rumen fill. To simplify that, basically, fiber that's not digested fast enough in the rumen accumulates in the rumen and cause a fuel effect that inhibits cow consumption. Consequently, this may lead to lower milk production and hinder the establishment of high forage diets. Just to exemplify that, many years ago, Oben Allen reviewed literature data, and they highlighted that for every 1% unit increase in NDF digestibility, either in vitro or in situ, could lead to 0.4 pounds per day greater intake of dry matter, and 0.55 pounds per day greater fat-corrected milk. Later, Jung and co-authors review the same aspect, but in diets containing more than 40% corn silage, and observe a similar response. So basically, we are certain that some of those indicators really translate into greater performance. But it looks like from some recent data that is not just associated with intake. Perhaps there is more to that. Okay, I'll try to go through this in the next slide just to put together a new theory behind this effect, but this may not really represent 100% of what we know. Okay, and I also think that there is still a lot to learn about this particular area. So in order to do that, we summarize data from 4 different studies. They are highlighted in the first column. And basically, in order to use fiber digestibility as the indicator, we use studies that compare sorghum and corn silage. I will not be discussing sorghum and corn silage today. It's just to exemplify the fiber digestibility effect. So all 4 studies, cows were fed exactly the same diet, but varying in sorghum and corn silage. So basically, when we go through the other 2 columns, intake and eating time, this data is presented as the percentage that cows fed sorghum silage had related to cows fed corn silage. So just to exemplify that, if we use the first row, Grant et al. 1994, cows fed sorghum silage consume 88% of cows fed corn silage consume. At the same time, cows fed sorghum silage spend 120% of the time the cows fed corn silage is spent. So if we put that into a perspective, you will see that for all 4 studies, cows fed sorghum silage, and in this case, cows fed less digestible fiber, they had between 85 and 95% of the potential of intake of cows fed corn silage-based diets. At the same time that they ate less, they spend between 5 and 20% longer eating. So what does that mean? And I don't really know exactly what does that mean, but what I understand from those particular numbers are either those cows were sorting, and because of that they spend more time to eat less, or they just took longer to eat, and consequently they probably spend less time resting. And this is very important because if we try to compare that with another data set, not related to those trials that we just presented, you will see that for every minute that a dairy cow spends eating, less performance they have. And this may be related to several factors that this particular results are not associated to fiber digestibility only, but perhaps they may be sorting, not resting, or there are any other constraints in the diet that lead to that factor. So now that we understand the full effect of fiber digestibility to a dairy cow, what are the options that we can consider to improve fiber digestibility? One of the options available is the use of more digestible hybrids. One example will be brown midrib mutant hybrids. Basically, brown midrib hybrids, they are a natural mutation that reduce the lignin concentration in plants. And to my knowledge, they are available not only in corn plants, but also in sorghum and per millet. And I believe they are available commercially for all those different forages. And what is very typical of the brown midrib is that because they have lower lignin, they are more digestible. So the question that we always receive is, do improved digestibility outweighs lower yields? And that's a very complex question. And the answer varies depending on the dairy and several other factors. Okay? I'm not going to discuss yield today, but I will anticipate to you that most hybrid trials around the country show between 8 and 12% lower yield for BMR. Well, Luiz, but you are showing a lot of lower yield there. Sometimes I cannot afford losing inventory. So what do you think about that? Well, if you cannot afford the inventory, perhaps this is not the best option, or perhaps you could incorporate that into some of your fields and feed your high producing cows, not to your entire herd, okay? But before you make this type of decision, what exactly brown mid-rib hybrids can do in the diet of lactating dairy cows? So we ran a meta-analysis some years ago, and from that particular meta-analysis, basically we compile all the literature evaluating different hybrid types. And in this particular comparison, we compare diets containing BMR corn silage versus diet containing control corn silage. And control corn silage could be conventional hybrids, dual purpose hybrids, or an isogenic line to the BMR being tested. So basically those would be hybrids typically fed to dairy cows. Okay? And I think when we look through nutrient composition, something that's very important to highlight is lignin is lower, almost a percentage unit lower than conventional hybrids. So this is the point in favor of BMR. And because lignin is lower, in vitro NDF digestibility is also greater, which basically is a reflex of the lower lignin and consequently greater fiber available for digestion. Potential downside is the starch percentage of as a percentage of dry matter. Sometimes some of the BMR hybrids, they have a slightly lower starch than the conventional hybrids. But what does that mean in terms of milk production? So in this particular slide, we are presenting what was lactation performance for the control group. In this particular case, cows that were fed conventional corn silage, dual purpose corn silage, the isogenic line versus the BMR. And then we have a column with what was the benefit or downside of feeding BMR to those cows. And basically, I think the response is very consistent across research studies. And cows fed BMR corn silage have greater intake, which leads to greater milk production. The downside of that is slightly lower fat concentration, even though there is no change in milk fat yield. And another benefit would be greater fiber digestibility, both rumen and total tract. And I think this may be changing the milk urea nitrogen levels highlighted here as MUN, probably because BMR may slightly improve nitrogen utilization. I believe there is a need for more research in that area, but that's what it leads us to think based on the MUN data. And another downside would be the total tract starch digestibility highlighted as TTSD here. And what I think is going on is because of the greater intake, the amount of diet passing through the gastrointestinal tract is higher. So it passes too fast from rumen and the small intestine. And because of that, it impairs digestion of starch. Alternatively, some BMR hybrids had a more flint genetic line. In flint hybrids, they typically have a slightly lower starch digestibility. So consequently, the combination of those two factors in this data set may be slightly compromising total tract starch digestibility. It's also my knowledge that some of the companies that commercialize BMR hybrids, they now have Flory genetic lines for brown midrib. And this may be of benefit as highlighted by some recent lactation performance trials in the Northeast. Okay, Luiz, but if I don't want to use BMR, how can I really use those indicators to select for hybrids? And here what I want to do is I want to tell you that UNDF may be a very good indicator, but you need to be careful with that. I'm not against UNDF. I think it's a great assay. From a laboratory perspective, I think it's outstanding because I don't have to run lignin, for example. But UNDF is typically reported as percentage of dry matter. And in my opinion, for selection of hybrids, it should be reported as percentage of NDF. But why do you think that? Well, let's use this image as the example. So let's assume that we have two corn plants. the one on the left with less starch, as you can see, less kernels, and the one on the right with more starch. If you have more starch in your sample, you will naturally reduce UNDF, just because you have more starch as a percentage of dry matter diluting UNDF. Always, but if there is less UNDF, it's more digestible. Not necessarily, because as you analyze the fiber digestibility, probably fiber digestibility will be the same, because the UNDF affects fiber digestibility primarily if you have the same amount of NDF. If you have less NDF, because of the dilution, UNDF may not be a good indicator. So if you want to use that as an indicator, which I do think is good, make sure you either find UNDF values as percentage of NDF or you calculate those. Now we'll shift gears to another important management practice for fiber digestibility, which is high cut corn silage. And this is a very hot topic this particular year, because tonnage seems to be very good across the Midwest. So a lot of people are considering to increase cutting height for corn silage. And if we use those two first corn plants to explain why, you see that when you increase from a normal cutting height, here highlighted is 8 inches, but it may vary between 8 and 12 inches depending where you are, maybe a little bit higher. And then high cut may be 16 or maybe even up to 24 inches. Right? So if you think about that and we look what is below the red line, which is basically below what you are harvesting, you'll see that the only thing you are not collecting by increasing the cutting height is the bottom portion of the stover of the corn plant. And that's where the vast majority of lignin is. So basically you should expect to harvest less lignin and consequently have a more digestible material. And this is already well established. But the question I want to answer today is, even though there are many studies that evaluated cutting height for corn silage, is there any comparison across multiple studies that can help us to understand what to do? And that's what we try to do. So in this particular table, we have first different parameters that are affected by cutting height, then the number of treatment means that were utilized. And the number I want you to pay attention today is what I highlighted as the slope. And what the slope here shows is what is the effect of increasing 1 inch of cutting height on these different parameters. So for example, if I think about dry matter, for each inch that I increase on dry matter, for each inch I increase on cutting height, I should expect 0.2 percentage units greater dry matter. This is also true for starch, basically because you are diluting the amount of starch by less forage material, because you are just eliminating the bottom part of the plant. Consequently, you have slightly less NDF and obviously lower lignin concentration. And this is of particular importance because this leads to greater in vitro NDF digestibility. But remember, if you are harvesting at higher cut, you are abdicating of some material, and this affects the total dry matter yield per acre that you can collect. So the question I have is, Luiz, can I use these numbers you are presenting today to try to generate some numbers to make my decision easier if I should or should not use that? And that's exactly the point with that. So we got one of our previous studies where we did measure corn silage at 6 inches and corn silage at 24 inches. So those first two columns, they are a real experiment where we collected several samples and analyzed that. And the third column is a simulation we use using the slope numbers I presented in the previous slide. So basically what I did is, I know that the difference is 18 inches. If I go back one slide, I know what changes per inch. So I know that if I multiply the number by 18 and I add to the silage here that I cut at 6, I can simulate what is the 24. And basically you can see when I compare high cut real versus high cut simulation that the numbers are very close. So perhaps it could be a tool to be utilized in order to make some decisions about increasing cutting height or not. Hopefully soon it will be available in a spreadsheet format so you can download for free and utilize that in the University of Wisconsin website. Now we're going to shift towards starch, and I promise that I have less things to talk about starch today than about fiber. And the most important indicators about starch quality in whole plant corn silage is first, starch concentration. Obviously, as you saw the impact of starch digestibility on total digestible nutrients earlier this presentation, you should expect that starch is very important. After that, we have a starch D, analyzing the lab. And for corn silage, we pay a lot of attention for corn silage processing score, or how well broken are the kernels. We're not going to really cover this topic today, but it's something that you should always consider. Okay? And regardless of having greater starch digestibility, or greater corn silage processing score, they all should lead to greater energy density of the material, which could improve milk yield or feed efficiency by maintaining very high yields, but having cows consuming a little bit less. So this is just a simple highlight of the potential effects of whole plant corn silage starch digestibility. So in the bottom left corner, you will see the total track starch digestibility varies between 80 and 98% of the starch. And these are the parameters we believe play a major role. First, kernel particle size or kernel breakage. If you don't break kernels, everything else is harder. Okay? After that, the time you allows it to ferment. That's the topic we'll briefly cover today. And then after that, kernel maturity and endosperm properties. Late maturity and faint hybrids are less digestible. And last, there are mentioned two additives, but in reality, we are still waiting for a very good additive to improve starch digestibility in the silage. Maybe there are some in the diet, but not necessarily in the silage. So the point I want to make today is that silage fermentation increase starch digestibility. And this review manuscript by Kang and co-authors, most studies available in the literature highlights that as fermentation progresses, starch digestibility goes up. And basically what's going on is bacteria that ferment silage, they have the ability to produce proteases, which are enzymes that degrade proteins, and they can degrade prolamine proteins. Prolamine proteins are proteins that surround starch and inhibit starch digestion either in the rumen of dairy cows or in the small intestine of dairy cows. So consequently, by having silage ferment for longer, there are greater potential for some of those proteases to break down prolamines and make starch more available. Alternatively, there are some proteases inside the corn kernels. And those proteases, they are typically activated at low pH. And as you all know, corn silage reach pH 4, sometimes even 3.5, right? So as pH starts going down, some of those kernel enzymes are activated, and they also break down some of those prolamine proteins. So the question we had some years ago was, can we use storage length as a tool to attenuate the negative effects of starch digestibility in late maturity corn silage and hybrids containing a more flint or vitreous endosperm? So basically our study was designed to answer the interaction question. Silage fermenting for longer, is it better for all the different case scenarios? So the first answer I have for you is yes, it improves silage digestibility in every single case scenario, but no, it does not attenuate the effect, okay? So here in the blue line, we have silage harvest at 2/3 of kernel milk line, and in the red line, we have silage harvest at the black layer stage. And basically you can see that starch digestibility over time, and we went from 0 up to 240 days of fermentation, starch digestibility keeps going up, but you can see that starch digestibility was always greater for early maturity. Perhaps if you're harvesting early and late maturity at your dairy, a wise way of mitigating this issue would be first, feed the early maturity corn silage, and then wait to feed the late maturity corn silage later on as it increased digestibility a little further. Unfortunately, this is also true for hybrid types. We compared 3 different hybrids, a leafy flory hybrid highlighted in the top right picture, then a dual purpose hybrid in the middle, and a brown midrib hybrid that had a very flint endosperm in the bottom right picture. So as you can see, the top kernel has a lot of white starch, right? Basically it's very digestible, and the others have a lot of this yellow fraction, which basically is a combination between starch and some prolamine proteins. And similar to maturity, you can see that the hybrid that was more digestible early on was still more digestible at the end of the experiment. Okay, so again, if you have multiple hybrids varying in starch digestibility, perhaps to feed the more digestible first and the less digestible hybrid later may be the best option. So in conclusion, there are many factors that can alter the nutrient digestibility of whole plant corn silage. We try to go through some of those today, even though there are many others that we all should pay attention while harvesting silage. And it is our opinion that harvesting practice are the most important factors to improve digestibility. Good processing, good maturity, good choices. Okay? Also important to highlight that if you have enough inventory to allow feedstuff to ferment for longer in the silo or to increase cutting height at harvest, those may be viable options to your dairy. However, I want to also highlight that inventory planning is required if you decide to do that. With that, I would like to thank you for your attention, and I will be glad to answer any questions you may have. Here are my contacts. Feel free to send me an email or a question. I'll be glad to reply as soon as possible. Thank you very much.